Hello everybody, welcome back to another video. This is Kenneth, and today I'm gonna be like continuing a three-part like protein sort of series, right? Where we had a Burum starting with bacterial transformation, and then I talked about like protein chromatography and like separation. So you may want to give those a watch before watching this video, unless you understand like how to separate a protein from a bacterial cell. And if you do understand that, that's good. And also, this video could be standalone if, say, you just had a protein like sample lying around and you just needed to test it and see like what kind of verify what kind of protein it is, because this is kind of for like verifying the protein in our example, uh, and this can be helpful for identifying proteins as well. So this is protein electrophoresis. Um, and you can see these steps might be carried out by a lab technician. But um, so first, what I'd like to say is this can be a little complicated. So if you need to like reread or look up some resources, maybe I'll put one in the description of the video. So, but yeah, let's get into this. So this is called like SDS-PAGE protein electrophoresis. And if you've ever heard of gel electrophoresis, which like for DNA, where the polarity of the DNA like makes it travel through the gel and then smaller DNA moves further. This is similar, except it's for proteins. And in gel electrophoresis, like DNA gel electrophoresis, the like gel is horizontal or like parallel with the like table or surface. And in protein electrophoresis, it's like vertical. Um, so it's like kind of standing up, and you have to insert the protein vertically, and then it'll like move down into the gel. But first, it is called SDS-PAGE, and why is it called SDS-PAGE? Well, this type of electrophoresis separates proteins based on their molecular weights, and the reason it does that is because proteins can have vastly different polarities and like sizes, right? Uh, so separating proteins based on polarities or any other sort of thing, it won't necessarily give you a clear like result or indicator of what you're looking for, because like any proteins can have the same polarity, like multiple proteins can have the same polarity, but there's a less likely chance that two proteins have the same molecular weight, um or like I guess amino acid count, just because proteins are more unique in that way. And so this leads to it being more useful to separate them based on molecular weights than like charge, right? So SDS is sort of a solution, and you can look up more about it. But basically, what it does is it unfolds the protein into their primary structure, so like the chains of amino acids, and it applies a negative charge onto the protein. And the reason is so that the proteins aren't separated by charge during the actual electrophoresis. So the charge of every protein being tested is the same, which is kind of like a control, right? Um, and the negative charge is used so that the proteins they migrate based on molecular weight, right? And even it's noted here that that wouldn't happen if the charges were not uniform. Now, page is actually the name of the gel, and that's a neutral thing. And it has pores in the surface that the proteins can migrate through once an electric field is applied. So basically, page is the gel itself, and the proteins move through the gel. So, um, if you look back to the protein uh, chromatography video, I'll explain like what this GFP is. But basically, that's a protein that we're doing electrophoresis on. So just think of like instead of GFP, but I'll say GFP, you can think of like any protein you want, really. So our GFP or protein samples are loaded into like a tube, and then a buffer solution is applied to them, and the buffer contains the SDS, which is crucial for this whole process, and that's also why the buffer is added to the sample. And so the SDS, like we said, it changes the proteins into their primary structure, and then it applies a uniform charge. And it also helps the proteins sink in water. So very important, vital for this process, okay? The protein samples are then placed in a 95-degree heat bath to make sure they're denatured and in the released or primary structure that we need them to be for this electrophoresis. And if you think back to biology, or if you have any experience with this, you'll understand that high temperatures will denature these proteins. So the tubes are cooled, and they're just flicked to make sure that all the things are mixed together and reacted, which is also important, because if the things don't react, then the proteins will be no different than they were before adding SDS, right? So in our example, or in our class, the second lane of the gel was loaded with precision plus protein standards, but in your case it may be the first lane. And what this does, it's kind of like how, well, let's think about to like DNA gel electrophoresis. There's a ladder which shows like certain amount of base pairs, right? You know the base pairs for that ladder, right? And then you can correlate those to match with the bands of your like tested DNA sequences. And this is similar. So this has like known molecular weights. The precision plus protein standards, when it's run through the gel, it shows known molecular weights, and we know them, so we can compare that with our unknown samples, which are the proteins we're actually interested in, to determine or approximate the molecular weights of our tested proteins. And this is the whole point of the lab, or the results, is to know that what the molecular weight our protein is, and confirm that that should be the correct molecular weight, or verify that that is the known molecular weight for the protein we expect it to be, or it can help us identify a protein if we don't know what it is. The samples you make are then added into subsequent lanes, and this is so that you can compare them with the standard lane, because obviously they still have to run through the gel, just like the standard lane, to be able to compare them. And basically, it's like a complicated process to load the proteins actually into the lane. And it's much better explained with an actual example. So I'll link a video to that below in the description, and I would take a look at it if you're unfamiliar with the actual lab sequence of inserting the proteins into the gel. But otherwise, let's move on to this. So how do you analyze the gel results? So after it's run, you place the gel on a light box, which illuminates the bands on the gel. And in our case, this is sort of like maybe a college level lab, but in an actual laboratory, I'm sure they have more advanced techniques than this. But in like a college level lab, we take a ruler and place it besides the precision plus standards lane, and we measure the length of each protein traveled from like the origin of the gel. A picture is taken, or we just record the notes, or the lengths, right? So we record the lengths that the precision plus standards lane, all the bands in the lane traveled. And we also record the length our sample traveled, of course, but that's gonna be just for comparison. So once we have the lengths, we plot the data from the precision plus lane, which we measured and noted, onto a logarithmic graph. So it should be relatively linear in our case, and then we draw a line of best fit. So what this is doing is allowing you to estimate a distance traveled in the gel for any molecular weight, because the Precision Plus standards doesn't have every single molecular weighted protein, because then it would kind of just be a straight line and you wouldn't really be able to tell in the gel like what goes with what, right? So you kind of have to, in our example, in like a college level class, you'd be taking an estimated guess and using a logarithmic graph to draw a line of best fit. And then once you have this line drawn, you also have the length that your protein sample traveled. And so what you can do is, well, you can compare that length with the estimated length of the Precision Plus standards, and then like draw a line across the graph to match that to the estimated molecular weight. And if you're successful, you'll see that if you know your protein sample and you know what weight it should be, you'll see that it should be correct or very close. And if you don't know the protein, then that just gives you an estimate of the molecular weight, and you can use that to try and determine the protein through some other method. That's pretty much the basics of this, but I'll definitely bring up some pictures to help visualize this. So give me a second here. Okay, so I kind of brought a picture from one of my friends here, and this was from our lab data in our class. And so what we have here is a line of best fit, and the points were the data that our Precision Plus standards lane gave. So for example, the Precision Plus standards gel or band that traveled 5 millimeters in our gel was about like 250 kilodaltons, which is the unit that our molecular weight for the proteins is measured in. And then, for example, at 9 millimeters here, it was about 150 kilodaltons for, and this is for the standards lane, okay? I just want to make sure that's emphasized. And then using that, you can see that although these points are like a little off path down here, we did draw a line of best fit, a linear one, for pretty much estimating like molecular weight for any protein based off the distance it traveled. And so in our example, right, we have our GFP protein, and this could be any protein, but in our gel, it traveled about like 24 millimeters. So we go up to the line, and then once it hits the line, we travel to the left, and you can see this is supposed to be like tens. So like this would be 20, this 2 here would be 20 kilodaltons, and this 3 would be 30 kilodaltons, and the 10 would be 100. And so this is about like 29-ish kilodaltons, and the GFP is like 26.9 kilodaltons in real life. That's the like measured weight. So you can see it's very close, and so it gives us a good estimate of what the protein is, or a good like verification method. That's pretty much all I have for this video. If you have any questions, please make sure to leave them below. And this is just an important skill to have for like lab techniques, of course. And that's it for this video. I will link those things I mentioned below, so if you want more clarification there, make sure to check the description. But otherwise, thanks for watching, and please like and subscribe if you enjoyed the video.